We sure can.
And bleep it out.
And YouTube may do that for us, although we didn't say that this was expressly content made for children, did we?
You are live, Jillian.
So I'll leave, huh?
But wait a second.
Yeah, it looks like it's going. Do you want to get started?
I think it's a different URL though. Okay. Yes. So I closed the YouTube channel right now because it's a little bit behind and you would always like hear that in the background. Okay, Alex, can you hear me?
Yes.
Cool. So I think we just start. I'm going to introduce you first.
Great.
So when I did my background research, I found a blog post that I actually had written in 2016 when I went to a conference. And there was Jessica Mark Welch was there, and she was presenting some really like mind-blowing work on plaque, dental plaque.
Mm-hmm.
So, and then I was like so fascinated there that I would write a blog post about it. And I even had a quote from her where she compared sequence-based human microbiome studies to bulldozing a hectare of coral reef, then mixing everything up and then trying to find or describe a clownfish anemone association. And that's how she tried to sell her way of like, when she showed some pictures of her lab where they took a small piece of plaque from human teeth. And I think that was most probably your work as well. And you use different color probes that would bind to known bacterial sequences at the real locations where they actually are sitting in the plaque, and then visualize it in 3D with this like classy fish method. And I was really excited when I realized that's you. So that was Alex Valm's work, who's now an assistant professor at SUNY Albany. And his talk today is entitled Structure and Assembly of the Human Oral Microbiome Across Spatial Scales.
Great. All right. So thank you, Letitia. It's absolutely a pleasure to be here. So if I try to share my screen, it says it's disabled.
Try again.
Let me try again. Yeah, now I'm able to.
Yes.
Okay, great. So I'm absolutely delighted to be giving a seminar in this series. So I recently discovered this series through Twitter, I think. Finally, something good that's come out of Twitter for me. And now I've been going back, and because you can look at them on YouTube, it's just fantastic. So I'm very pleased and delighted to be a part of this seminar series. So, right, so I will just dive in, so to speak, with my talk. Now, I always like to start my talks off with this image, and many people may have seen this before. So this is the first direct observation of bacteria. I call this the Human Microbiome Project, circa 1690. So these were drawings made by Antony van Leeuwenhoek, who made a microscope. Actually, it was a single lens microscope, so more like a magnifying glass. And he observed bacteria, and he made these drawings. And I didn't know this until I was in grad school. The source of this bacteria was actually his mouth. So he scraped his teeth, and he was looking at his dental plaque, and he saw these bacteria. And a number of things just jump out at me from this image. So first of all, what's remarkable is the realistic drawings. So from looking at these drawings, I know what organisms, or I have a pretty good idea of what organisms he was describing. So this is a spirochete. These are abundant in the oral community. They're treponines. This boomerang-shaped organism is probably Leptotrichia. The cocci are most likely Streptococci because they're the most abundant of all organisms in the mouth. As well, they're pretty large for coccoid cells. And this one here, which he's drawing moving, is probably Selenomonas because it's one of the few organisms that are flagellated in dental plaque. And so besides the fact that he actually saw these things, what's really interesting about this is that, first of all, our first direct observation of bacteria told us that they live in the human body. Van Leeuwenhoek predated Robert Koch by over 100 years. So Van Leeuwenhoek made no association between disease and bacteria. So bacteria in the mouth was part of normal health. As far as we know, Van Leeuwenhoek had healthy teeth and gums. Oh, sorry. And lastly, what's really interesting, if you really think about this, so Van Leeuwenhoek is drawing this drawing. He's trying to classify these organisms, but if you can imagine yourself in his place, he must have seen all these organisms living together. And so our very first observation of bacteria told us that they live not as monospecies in culture, but as mixed communities. And somehow we forgot that immediately after this initial discovery. So I know that most people in this audience are microbial ecologists, and so I won't focus too much on disease. I will really focus on the ecology of the oral microbiome, but just a little background here. So let's see if I can make my pointer. Okay, hopefully you can see my my mouse, but in the mouth, we recognize that these microbial communities are responsible for maintaining health. They are also involved in disease. And so what's really interesting about the mouth is that there are two major diseases that are mediated by microbes, dental caries and periodontal disease. Both of these diseases are caused by organisms that are present in communities in states of health. So what we understand is that we consider a healthy microbiome or a healthy microbial community to be a state of eubiosis. Something happens to this community in which it undergoes a shift, and we call this shift dysbiosis. And what happens is a subset of organisms that are present in states of health become overabundant in the community. So the relative abundance, and actually the absolute abundance of certain species changes. Certain species become more abundant in the mouth. I'm going to stop using my mouse. And coincident with this is involvement of the immune system. That leads to pathological changes, like the damage that occurs to the tissue, including loss of the soft tissue, loss of bone, and eventually loss of the tooth if it's not treated. There are a number of things that we could dive into in this model, but what I want to draw your attention to is actually what's represented in this model. If you step back and look at it, are different spatial scales. So on the one end, we have this sort of macro scale, this scale on the order of centimeters, let's say, it's the relevant scale for the different structures and tissues in the mouth. We know that different parts of the mouth, like the tooth above the gum line versus the tooth below the gum line, support different microbial communities. So there's that scale, that centimeter scale. On the other end, completely other end, is the molecular scale, the nanometer or micron scale, which are the molecules that are involved in either a direct association with the host or between the microbes. And then what's in the middle between these two scales is what I'm going to call the mesoscale. And that's the scale of these, the microbial cells themselves and the structures that they form. And so I just want to say that in all of these types of models, the microbes are drawn like this as a mix. They're sort of well mixed together as if they're planktonic, and we know that that's not the case. So I'm going to make a case for the importance of spatial structure, especially at that mesoscale. So that's what my lab is interested in. Here's a sort of summary representation of what I just said. So at these different scales, we can use different techniques to study the microbial community. So at that macro scale, like you mentioned, Jessica Mark Welch's analogy, we can use sequencing. That can tell us what is the community composition in one part of the mouth, the tooth, versus another part of the mouth, like the tongue dorsum, for example. On the other end of spatial scale, for the most part, it's biochemical assays, genetic screens that can identify the molecules that are involved in the metabolic and physical interactions of individual cells. At the mesoscale, I'm going to argue that what's needed to study this scale is imaging, that's microscopy, and maybe we have sort of not given enough attention to this spatial scale, and I would guess that the reason for that is because the tools are just underdeveloped for imaging at this scale, even though we know that this scale is important because this is the scale of the biofilm, and we know that biofilms have emergent properties. So we need to study the spatial structure of microbial communities at that scale. Here, I'm just going to hammer this point home, that spatial structure is important in understanding the function of microbial communities. So these are two different communities that are associated with, let's say, mammals. They're, on the left is a large intestine from a mouse. That's actually from Mark Welch and others. And what's important here is that you see that in the intestine, we understand that there is a physical separation between the normal microbiota and the host epithelial tissue, and that this separation is important. It's mediated by a mucus layer, and if bacteria are able to get through this mucus layer and make direct contact with the epithelial cells, that elicits an immune response. It's a pathologic immune response. On the other hand, in the skin, here's an image of a human hair follicle. We understand that, or we're learning that the microbes that are present on the skin are actually present not only superficially, but deep within glands and hair follicles. So this is a hair follicle. The yellow is the shaft of the hair. The cyan color are the stem cells that support the hair itself. And what's represented in magenta here are bacteria. And if you look even closer, you'll see there are fungi that together form a biofilm associated with this hair. And we understand that this biofilm makes intimate contact with the immune cells, especially the Langerhans cells that are present in the skin. And the hypothesis is that this direct contact is important for training, proper training of the inflammatory response. So spatial structure is important in understanding the function of human-associated microbial communities, especially whether they're going to promote health or disease. Again, DNA sequencing tells you what organisms are present. Biochemical assays can tell you, have informed a general hypothesis for the structure of dental plaque. So what's shown here on the left are these remarkable assays called coaggregation assays that have been performed for thousands of different species that have been isolated from the human microbial community. So we who work in the human microbiome field, or especially in the oral microbiome field, are very privileged because actually at this point, about 70% of the species that we think are normally found in the mouth can be cultivated in the lab. So cultivate these species separately in different liquid media, you mix them together. If they clump and sink to the bottom of the tube, we call this coaggregation. And if we can inhibit this with specific molecules, then we can learn something about the nature of this interaction. If the organisms are mixed and they stay well mixed in the tube, then we conclude that they're not directly interacting physically with each other. And so this kind of binary assay has been performed, like I said, for thousands of combinations of microbes. And that led to this general hypothesis for the structure of dental plaque, where you have the tooth surface, which is coated with a salivary pellicle. So these are, for the most part, glycoproteins that are present in saliva. And then there are a subset of organisms that are able to bind specifically to this salivary pellicle. So these are organisms mostly of the genus Streptococcus, but also Actinomyces and some others. And what's important now, we get into the ecology of this community. We recognize that there's an ecological succession, or at least that's the general hypothesis, that the salivary pellicle is present on the tooth. And let's say you brush your teeth, you remove all the bacteria just for the sake of argument. Let's say we remove all the bacteria, even though that may not be possible. What happens is the early colonizers are able to specifically recognize molecules in the salivary pellicle. Then in time, if this biofilm is not disrupted, other organisms can come in and bind to these organisms, and so on through time. And so you get these, what we call the early colonizers and the late colonizers. What I want to draw your attention to in this model is this organism here, this one organism called Fusobacterium nucleatum, which is hypothesized in this model to reside sort of in the middle, and it functions as a bridge. So unlike most of the other organisms that are able to make one or two physical connections with other species, Fusobacterium nucleatum has demonstrated the ability to bind specifically to many different species. And so, and importantly, both the early colonizers and the late colonizers. So there is a hypothesis that this organism functions as a bridge to bridge the early and the late colonizers. Why is this important in the context of human health? These early colonizing species are the ones that are associated with periodontal health. The late colonizing species are the ones that are associated, are the ones that become overrepresented in states of periodontal disease. So this comprises the ecological plaque hypothesis. And it pains me to put this on here, but I have to remind myself to say this. So I tell my students in my microbiology class that this is a bad hypothesis, or at least it was a bad hypothesis when it was proposed in its last iteration around 2010. Why was it a bad hypothesis? Because it couldn't be tested. The tools were not available. So do I have it here? Okay, right. So so my lab is interested in testing that hypothesis. And we recognize that we need to develop tools to do that. So we have two focus areas. One is the development of technological tools. The goal is to expand the number of fluorescent labels that can be distinguished in a sample. And then we have a biological tool development where we are developing an in vitro biofilm model that we can use to test these specific hypotheses about the systems level spatial structure of communities. So in terms of that first tool, the technological tool, as a first approach, we make use of commercially available technologies. So we use confocal microscopes that are capable of spectral detection. Basically, these point scanning instruments, instead of sending the light through a filter, they sample the light at different wavelengths. So there you have a spectrometer attached to your microscope. You record the full spectrum at every spot, every point in the image. That becomes a pixel in the image. And then we use algorithms to identify what are the fluorescent molecules that were present in that pixel. Our early work involved the use of those commercial instruments as well as a combinatorial labeling strategy, which Letitia talked about. So we reasoned that we could further expand the number of identifiable species by labeling microbes with specific combinations, giving almost a barcode approach for identifying organisms in an image. And we did call this ClassyFish for combinatorial labeling and spectral imaging. Right, and so we first set out to prove proof of principle that this could work. So we did this with E. coli. We made an artificial community of differently labeled E. coli. So these are all the same strain of E. coli, but they were just labeled in separate tubes, such that each tube got a specific binary combination of fluorescent dyes in a fish labeling experiment. We mixed this together, like I said, to make that artificial community, that mock community. We spotted it on a slide, and here's what the images looked like. This is the image after we apply that algorithm for spectral identification. But remember that every cell is labeled with 2 fluorophores. The human eye can't really distinguish all 28 of these different binary labels, but that's fine because this whole process is computational. Once it gets on the microscope, it's a digital image. The algorithms that are used to identify the fluorophores that are present are all computational. We wrote our own software code to identify the cells in the image and then identify the binary label. So we don't even need to look at these images. A computer can analyze this for us. And we do find that all 28 were present in the right proportion. So after that, we moved on to a biological proof of principle. And so around this time, the Human Oral Microbiome database was released. We mined this data to find the most abundant genera in the oral community. We identified 15 genera that were highly abundant and highly prevalent in the oral microbiome. And we decided to go after those genera. It turns out that those 15 genera cover between about 80 to 85% of the community. So we designed 15 genus level probes. And then again, we made an artificial mixture. So here what we did was this, we acquired isolates, representative species of each of the 15 genera. We grew them separately in their own media, whatever they needed. Then we mixed them together in this artificial community. But we mixed them first, then we applied our fish probes. And so we're testing our probes. Our probes worked. They identified all 15 genera in the right relative proportion. And that was evidenced by, again, our software tool that we developed to identify. Now, the binary label actually means something. It tells you what the genus is of the organism. So we set out to apply this then to a natural community. We recruited volunteers to come into the lab. They flossed their teeth for us, handed over the floss. We scraped off the dental plaque off the floss and put it in a tube and did our fish labeling in a tube. So I call this semi-dispersed dental plaque. After we labeled that, we spotted this again on a slide. And what we saw overwhelmingly were these clumps, these multi-genus clumps of organisms that were scattered throughout our images. And we wondered whether these associations were present in the biofilm before we disrupted it with floss, or if they came together artificially in the tube while we were doing our fish experiments. And so we did some computer modeling to try and identify what we would expect from random associations. Nowadays, we would do this, I think, with AI, some machine learning approach. But this allowed us to identify all the statistically significant intergenus pairings present in the community. And so this allowed us to develop this network analysis. And In this analysis, the size of each node here represents the absolute abundance of that organism in the community. A line connecting two nodes means that those organisms were found to associate with statistical significance. And so what's interesting is that, as far as I know, this was the first network analysis of its kind that was done that was informed by spatial data at that nanometer or micron scale. And what I want to draw your attention to is Fusobacterium. So according to that early model, Fusobacterium should be here in the middle, and it should be connecting with many other genera. But in our analysis, it wasn't. It barely survived our p-value cutoff of 0.05, and only with 2 organisms. So that's something to keep in mind. So further development of our technology. So I mentioned that in our analysis of the oral microbiome, we identified 15 genera that covered about 80 to 85% of the community. It turns out that there are we recognize about 700 species that are present in the community. About 30 genera comprise the community. And in any individual person, in any individual host, there are about 100 different species that are present in the mouth. So we wondered, could we expand our technology and be able to identify 100 different microbial targets in any single image? And so we set out to do this. Some simple math told us that we needed a minimum of 15, but 16 individual fluorophores in binary combinations would result in 120 unique combinations, or 120 barcodes. And so we identified 16 commercially available fluorescent dyes that are well spaced throughout the visible spectrum, but are highly overlapping. And we used commercial technology. We used the same available confocal microscopes to acquire our images, but we developed our own algorithms for analysis, spectral deconvolution. So our algorithm, first of all, uses both excitation and emission spectral information. So rather than a spectral curve, now we have this sort of matrix representation of the fluorophores. And the second thing that we did was we used some a priori knowledge. So we knew that every organism in our sample would be labeled with exactly 2 fluorophores. No organism would be labeled with 1. No organism would be labeled with 3. So we constrained our algorithm to find only binary combinations. We said only binary combinations are possible in this image. And when we did that, what we found was that when the signal to noise ratio was very high, if the sample was very bright, our binary constraint didn't really improve the result. But when the signal to noise ratio was low, when the sample was dim, we had a significant advantage, and that's due to the noise that's present in fluorescence image. So our modeling told us that this should work. So we went out again, we made an artificial community, this time of 120 different E. coli's. We mixed them together in this community, spotted them on a slide. Here's a low power image. And again, we were able to identify all 120 labels, and they were present in the abundance that we expected based on how we made the mixture. Right, and so just a little bit of a foray now into how we're applying this technology. What I have told you about up to this point is our initial work that involved imaging disrupted biofilms. Of course, we would like to image intact dental plaque biofilms, and so we have a couple of methods for that. First of all, we're looking at, we have a collaboration with clinician scientists at the NIH, where they have healthy volunteers come into the lab. They wear this dental appliance in their mouth. On the side of the appliance are what we call enamel chips. These are actually pieces of other people's teeth. Usually they're third molars that people have extracted. They're stuck on the side of this appliance and people wear this for a day or so. Then these chips are mailed to us at Albany and we can do the imaging here. And what we see is that we do see mixed communities dominated by those early colonizers. So the magenta color here again are streptococci, the blue are Haemophilus. And so with this approach, we're able to make some qualitative assessment of the community. We do see only the early colonizers, not the late colonizers. And we saw some new structures that involved Rothia and streptococci that hadn't been observed before. Excuse me. So we also are interested in, of course, looking at teeth. So we're interested in, we do get teeth that are extracted from patients. Now, usually these are patients with periodontal disease. For some reason, healthy people don't want to have their teeth extracted and sent to us. I don't know why. But so what we see in these patient samples is that, again, we do see, as we expect, the early colonizer, Streptococcus, is most closely associated with the tooth surface, and the middle colonizing Fusobacterium, not in the middle, but actually spread throughout the biofilm is what we see. And so what we're currently working on, a huge thrust of my lab, is to develop the protocols for extracting teeth, preparing them for sectioning. These are hard tissues. So the tooth is similar to bone, but the biofilm attached to it is a very soft tissue. So we're working on protocols so that we can both decalcify and be able to section the tooth, but preserve the soft biofilm that's attached to it. Lastly, we're also doing what we call plaque smear imaging. And so this is an example where we have a volunteer, again, floss their teeth, and then instead of scraping the floss off, we very gently lay the floss down onto a microscope slide. When you do this, if you scan around on the slide, you'll see, disrupted biofilm everywhere. But every now and then, you see these large structures that look like they may have been intact and present in the biofilm before it was disrupted. And so, again, our colleagues, Mark Walsh et al., call these structures hedgehogs. And what we see is that we think that this was the tooth surface and that this was the apical surface of the biofilm. At the tooth surface, again, we see the early colonizers. We think they function as anchors, Streptococcus and Actinomyces, for example. But surprisingly, we see these organisms again at the apical surface. And so many of these organisms at the surface are facultative anaerobes. And so we think that they're functioning to sequester oxygen, because in the middle of this hedgehog structure is where we find anaerobes. So yellow are Fusobacterium, the cyan color are Porphyromonas. Those are both strict anaerobes. And so we're updating that ecological plaque hypothesis, where it's not just a case of linear succession that occurs in the community, but there's a remodeling of the community as organisms sort of remake their niche within the environment and make it suitable for other organisms to co-localize with them. Right, so that's part one. Part two now is our biological tool development. And again, like I mentioned, we are interested in testing hypotheses about the systems level spatial structure of these microbial communities. And so rather than just observe the structure that's present in a host, for example, we want to grow communities and be able to perturb them. So for that, we borrow a remarkable method that was developed by early environmental microbiologists called the microcosm. So I probably don't have to explain this to most of the audience here, but I just find this fascinating, that you can go out into the environment and dig up some soil and put it in a beaker. You bring it into the lab, and what you've got is a mixture of microbes that were present in that soil sample when you collected it. You don't know exactly what's there, but it's a community. And in the lab now, you can control certain aspects of the environment, the light, for example, the water, for example. And so you have this sort of hybrid model where you don't know exactly, you don't have complete control over what's in the model, but you do have some control over the conditions. And so you can you can make specific perturbations and test their effect on the community as a whole. And so we borrow that technique. We didn't invent this. We were not the first to apply this to the oral microbiome, but we are trying to adapt this for imaging. So we have volunteers. We have volunteers come into the lab. They donate their plaque and their saliva to us, again with dental floss or spitting in a tube. And we use that dental plaque as an inoculum to grow a mixed species community. And we do this in microfluidic devices to mimic the flow of saliva over the surface. And in our early work, what we saw was that when we use a rich medium, we get a community that is not diverse. It's dominated by, in this case, streptococci. And this is a general phenomenon of laboratory culture. If you try to co-culture different organisms in a lab, the majority of the time, one or a few organisms has some very slight metabolic advantage under those laboratory conditions, and it's able to expand in the community before anything else can grow. With our dental plaque microcosms, what we found was that if instead of using a rich media, we used a very minimal media, in this case, 25% human saliva. Now, human saliva is already 90% water, and we add another 70, we dilute it 1:4 in water again. So we have basically water that we're growing these organisms in. We find a very highly diverse community. So in this fish image, all the gray cells are labeled with UB probe, which labels all bacteria. So we don't even know what these cells are. We don't have probes yet for them. So I think this is really strong evidence that there is metabolic cooperation in the oral community. Probably this is a phenomenon that is universal among microbial communities. But this result told us that we need to do some sequencing to find out what's present in our microcosm cultures. So here's our protocol. We bring in volunteers. They donate saliva and plaque. We take this and we sequence the initial inoculum. For the most part, we're doing 16S sequencing. Then we use the inoculum to inoculate microfluidic chamber. We grow our biofilms over time. Then we harvest them. We always do our biofilms in duplicate. One biofilm goes for classy fish imaging. The other biofilm goes for 16S sequencing. And so our initial results, told us that we are able to grow diverse communities in the lab. This was phenomenal. The day that this data came in was the best day of my assistant professor life so far. Actually, that's not true. When my two grad students told me they wanted to join my lab, that was the best day. But this was the best day in terms of data. So here's our donor inocula, the plaque and the saliva. If we grow, I didn't mention this, but we grow our biofilms, like I said, in human saliva dilute, or we have an artificial saliva media that we use. And in both cases, we grow very diverse communities. Of course, there are some differences between the in vitro community versus what was present in the initial inoculum. That's to be expected. But importantly, what we see is that this rust-colored band, that's Fusobacterium. That's the organism that my lab is very interested in. We still want to nail down this, this hypothesis of what the role is of Fusobacterium in organizing the community. Right, and so here's sort of hot off this is the most recent work that's come out of the lab. So we are able to grow diverse structures in vitro. In this case, we're labeling with 6 different labels. We see all 6 of these organisms present in the communities. We do see a large degree of heterogeneity. So from one biofilm to the next, even though they're inoculated with dental plaque that was collected from the same donor at the same time, we see these heterogeneous structures that form. And so the biggest thing we're doing right now is we're trying to understand we're trying to describe that heterogeneity and then understand what causes that heterogeneity. But just a little tidbit, we do think we're growing structures that approximate those hedgehogs in vitro. So we do see fusobacterium spread throughout the biofilm, and then those other organisms at the apex, and also at the, in this case, not the tooth, but the microscope, microfluidic slide surface. Right, and so here's a 3D rendering of that. So we're calling these potential hedgehogs in vitro. And we're, again, like I said, we're trying to understand that heterogeneity. And we do think that that stochastic events are involved in nucleation of these biofilms, and that's something that's been that's being realized by other labs as well right now. So so I'll just go ahead and show you our conclusions here are that our oral microcosms do recapitulate natural communities, the natural community that's present in the mouth, in terms of taxonomic composition extremely well. In terms of spatial structure, somewhat. We don't see that beautiful hedgehog structure, right? We see something that's more of a gamish of microbes. So we're working on trying to see if we can get that to look more like a hedgehog, or at least to understand why it doesn't. Secondly, we do identify what I'm calling obligate social taxa versus facultative social taxa. So I didn't point this out in the images, but what we find is that there are some organisms that seem to be able to live on their own anywhere on the surface of the functionalized surface of the microfluidic chamber. But then there are some organisms that we never see alone. They're always with other organisms. And so so we've developed this sort of lab terminology here. And interestingly, this recapitulates work that's already been done, but in this case, we're doing this in not with isolates, but with microbes that all came from the same donor. So in conclusion, here's our model. What we think is that our in vitro biofilms are somewhat stunted in their development. We think that stochastic events contribute early to biofilm development, and that intercellular interactions then contribute to the growth of these biofilms. What we're missing in our lab cultures are the microenvironmental inputs and any of the host immune factors, for example, that we think shape the mature plaque biofilm. Nonetheless, we do think that our model is a powerful tool to test hypotheses regarding the systems level spatial structure, especially these how these intercellular interactions are involved in structuring the community. So what's next? Again, because our microcosms, we don't have control over what goes in, we can't leave an organism out. We do want to test this fusobacterium hypothesis. And so there are a number of surface molecules that are known to mediate its interaction with other organisms. We'd like to see if we can inhibit these interactions, perhaps with neutralizing antibodies, for example. And so my last slide, of course, is the most important, where I acknowledge the work that was done. So Sahar Malek is my lab manager. She did a lot of the molecular work, the sequencing, a former lab manager, Sarah Araldi-Brondolo did the actual sequencing results that I presented here. Alex Lamas is doing the in vitro biofilm work. Ethan is doing the technology development. And I acknowledge my funding sources and especially the people that vote for and support these funding sources. So with that, I'm done, and I believe that we do take questions. Is that right, Letitia?
Yes, we do actually.
Great.
So thank you very much. This was a really terrific talk. I am honored to host you. I see that first we have on the YouTube channel, we have some questions popping in. So one question is, you briefly mentioned sectioning the teeth after decalcification. Do you section for the in situ images?
Yes, that's right. So, we have to section the teeth because with confocal imaging, we can only image about 100 microns in depth. And so the surface of the tooth is such that it varies over more than 100 microns. So we have to section the teeth in order to use confocal microscopy to do the imaging.
Okay, and are the in vitro hedgehogs, are they attached to the slide?
Yes, they're attached to the slide. We functionalize the slide with saliva. So before we add the inoculum, we coat the slide with saliva from the same donor, and that forms that pellicle. So the saliva will stick to the glass because of anionic or ionic interactions. And then we get our early colonizers, for the most part, will then bind to the slide.
Do you think the host plays a role in structuring these hedgehogs? So one side would be in contact with the host, with the tooth or the gum. Would that make it difficult in microcosm?
Yeah, actually we do. So if I can, I wonder if I can, I probably can't, let's see, I can do this. So we do think that the host is important. So what I'm gonna show here is unhide this. So I'll draw your attention to the right side here. So this is a electron micrograph of, in this case, this is subgingival dental plaque. So this is below the gum line from a dog. Dogs are somewhat unfortunately the best model we have for periodontal disease. They're not used that much. But in this early work, what you see is that here's the biofilm and here's the host immune cells. These are the shape of the nuclei tells you they're probably neutrophils. They're the proximity of those neutrophils to the biofilm, we think, suggests that of course they're involved in shaping the actual structure of the biofilm. So we do think that that's important, and we definitely don't have that in our in vitro biofilms. For if we want to include that, we need animal models.
How does your method help identify a dysbiosis? Do you have like spatial connections that can be interpreted as a healthy microbiome and connections that would indicate the start of a dysbiosis?
Yeah, that is fantastic. So that is the million-dollar question. Right, so ultimately that's our hypothesis, that the structure is involved. And that what we think is that there is so what the community recognizes is that there is there's a back and forth between the host and the biofilm and the environment, okay? And so behavior, oral hygiene, whether you smoke, for example, your genetics and your biofilm come together to either promote eubiosis or dysbiosis. And once dysbiosis starts, it's a vicious cycle. It sort of self-fulfills itself because the organisms induce inflammation. That inflammation then actually causes the biofilm to expand because it provides nutrients to the biofilm. So we would like to find out the golden ticket here would be to be able to find early markers of that that transition from eubiosis to dysbiosis. We think if we can find it early enough, we can stop it and reverse it. And so our hypothesis is that we can identify spatial interactions that might be early evidence of the transition. We haven't found them yet, but that's really what we want to do with our technology.
What is the variation of a healthy microbiome in teeth among patients? Is there a lot of individual variation, or is there a core that you find and could then like stick in a toothpaste or something?
Aha, yeah. So, okay, so I'm gonna tease up. So there's two major things in that question. So is there variation? Yes. Is there a core microbiome? Yes. So the answer is yes to both questions. I think that, you know, like all of microbial ecology, it depends on what level of taxonomy you look at, right? If you look at a very high level of taxonomy, if you look at phylum level, all humans have the same phyla in their mouth, okay? If you go down to genus level, it's very similar. When you get to species, you start to see variations. And when you get to strain, then you find highly unique communities in each person's microbiome. And I think that as far as I know, that is a paradigm that's true for most microbial communities that we've studied. Now, the second part is getting at probiotics, right? Can you make a probiotic toothpaste? Definitely people are working on that, right? So for sure, because these organisms are interacting with each other metabolically, biochemically, physically, there's a lot of room to search for probiotics. And we're not actively doing that, but there are labs that are looking at that. So I encourage you to look at those studies.
And maybe a last question would be, have you ever used somebody else's toothbrush?
Have I ever used somebody else's toothbrush?
Or would you like say no?
Right. I think I'm going to plead the fifth there. I'm not going to self-incriminate myself.
Okay. So yeah, thanks again for your really great talk. I think there are no more questions.
Okay, great. I should say I should have had my email up here, but if anyone wants to contact me via email, I'm at SUNY Albany, so you should be able to find me. It was really a pleasure to be a part of this, a pleasure and an honor to be a part of this series. So thank you.
It was an honor for us.
Great, thank you.
Cool.